we have seen several types of receptors in the skin for touch, pressure, vibration, pain, temperature. Okay? So now, how they convey or how they send the information to the spinal cord. I told you that we have several types of fibers, nerve fibers, and some of them can be activated by these cells, right? So, all of those 5 receptors, except the free nerve endings, use 8 beta fibers. So, myosin, melatonin, pacinium, graphene, and hair receptors all use A beta fibers. So A beta fiber is a myelinated fiber which is very thick and fast in transmission of the signals, right? Conduction velocity is between 30 meter per second to 70 meter per second. So it's very fast. And these fibers carry the precise type of signals, or the critical type of the signals, like precise localization of the touch, minute gradation of the intensity. So, a little increase in the intensity of the sensation can be felt because of the action of these fibers, okay? And rapid changes in sensory signal intensity. So, critical type of signals are conveyed through the A beta fibers. But free nerve endings use mainly A delta fibers, and some of them use C fibers. Okay? So, remember, we are talking about those 6 receptors. Receptors involved in touch, pressure, vibration. I'm not talking about temperature or pain. Okay? For those receptors that I started with, those 6 receptors, free nerve endings use mainly A delta fibers, and some of them use C fibers. A delta fiber is myelinated, C fiber is unmyelinated. The type of action potential conduction here is saltatory, remember? But in C fibers, we have point-to-point action potential conduction, right? So, the transmission in A delta fiber and also in A beta fiber is saltatory, it's very fast, but in C fiber is point-to-point, right? So it's slow. For pain, remember we have different type of receptors: thermal, mechanical, polymodal, silent, right? So you can see that thermal and mechanical pain is transmitted through A delta fibers. Mild. But polymodal receptors, those that respond to all of the stimulation, thermal, mechanical, and chemical, right? So these are transmitted through C fibers. Silent nociceptors also use C fibers for transmission of the signal. So again, pain receptors use both A delta fibers and C fiber for polymodal and silent receptors, A delta fibers for thermal and mechanical. And you know that A delta fibers are faster than C fibers. Yes. So if they have a painful stimulation, right? At first, at the same time, all of these receptors are activated, right? So, for example, mechanical and polymodal are activated. So what happens? The CNS first receives a very fast activation, very fast action potential, and after some milliseconds or after some seconds, it receives another signal. The first signal receives through A delta fiber. The second signal comes through C fibers. So there is one stimulation, one painful stimulation, but you feel two pains. A fast pain and a slow pain. So that's why we have two different types of pain. Fast sharp pain, slow chronic pain. Okay, fast sharp pain is transmitted through A delta fibers, but slow chronic pain is transmitted through C fibers. Okay? Slow pain is felt after 1 second. After 1 second of a stimulation. So when you have a pinprick, at first you feel the first pain, and after 1 second you feel the second pain. So we call this double pain. Right? Okay, what about temperature? Thermal fibers use A delta, actually are either A delta or C again. For cold, we have mainly A delta fibers. Some of them are C fibers. And for warm, we have C fibers. So C fibers are for warmth, for cold, some of them, right? For chronic pain, right? For tickle, for itch, for crude touch, right? Crude touch, I told you that touching the cornea can cause touch, and also touching the body, okay? Actually, is conveyed through two different systems. Those 5 specialized receptors that are very precise, and also through 3 nerve endings that are not so precise, okay? So, you should memorize all of these details, right? Okay, so we have seen the receptors in the body, the fibers. Now let's focus on the spinal tracts. So, for transmission of the signal through the spinal cord to the brain, we have 2 different pathways. DCML stands for dorsal column medial lemniscal system, and anterolateral system, spinal thalamic system. They have lateral spinal thalamic and ventral spinal thalamic. You should know these by the neuroanatomy, right? You have this. So I'm happy. So dorsal column medial lemniscal system is composed of large myelinated fibers, AD. But anterolateral system uses small myelinated fibers, maybe a delta. Okay? So smaller myelinated fibers for anterolateral system, large myelinated fibers for DCML. So this system is faster than this one, right? And spatial orientation of the fibers in the DCML system is very high. I will talk about that. But spatial orientation in the anterolateral system is low. The type of the signals that are transmitted in the DCML system are these: discriminative touch, or fine touch, or light touch, okay? Vibration, movement against the skin, position, finely graded pressure. But type of the senses that are transmitted in the anterolateral system include pain, temperature, crude touch, deep pressure, tickle, itch, sexual sensation. Okay? But I'm afraid you should memorize all of these details, you know? Yeah. So, you should remember all of the sensations that are transmitted through the DCML and all of the sensations that are transmitted through the anterolateral system. Now let's see the DCML system. Okay, I told you that beta fibers bring the information to the spinal cord through the 5 specialized receptors. right? So, when they come to the spinal cord, they are divided into two branches. A medial branch, a lateral branch. So this one is a beta fiber from Meissner, from Pacinian, Raffini, and all, okay? They come to the spinal cord through the dorsal root. They are divided into two branches, the lateral and the medial branch. Let's see the medial branch. The medial branch goes directly into the dorsal column and turn upward and goes up. Non-stop, without interruption, without any synapse. Okay? So the cell body is in the DRG, the process coming to the spinal cord, without any synapse, it goes up to the brain. Okay, we'll see what happens to this fiber. Then, the lateral branch make contact with neurons in the dorsal horn. So we have a lot of cell bodies here, so the fiber goes to different layers of the spinal cord gray matter. At each layer, they make synapses with different neurons, and those neurons may go to different locations or may serve different functions. Some of them turn back to the dorsal column and go up. Actually, most of them. Okay? But some activate anterior motor neurons and cause reflexes. Some others go to the spinocerebellar tract. So they go to the cerebellum. Right? So this is all that happens when a beta fiber comes to the spinal cord. So first, it has a medial branch, a lateral branch. Medial branch goes directly to the dorsal column and ascends in the system. Lateral branch makes synapses on neurons. Those neurons, those second order neurons, most of them come to the dorsal column and go up, right? Some of them cause reflexes and some go to the cerebellum, right? So now let's see dorsal column. The medial branch enters the dorsal column, right? So you know that in the dorsal column we have two different fascicles, right? Gracile, very good. Gracile actually collects information from the lower part of the body, like the leg, right? And the cuneate one from the upper limb and the trunk, right? Very good. So a light touch in the skin may activate Merkel and Meissner corpuscles. Okay? They sense the A beta fibers in the spinal cord, cell body is in the DRG, right? They come to the spinal cord, the medial branch enters the dorsal column and goes on in the spinal cord. Then fiber from the hand join these fibers and they also go up. But this one ascend in the cuneate fascicle, right? But this one was from the gracile, right? They enter the medulla. So spinal cord is finished. We have medulla. In the medulla, we have dorsal column nucleus, gracile nucleus, cuneate nucleus, right? So fibers end here. So the first synapse in the DCML system occurs in dorsal column nucleus, right? The first neuron in the DCML system is in the first neuron in the DCML dorsal root ganglion. Cell body is there, right? So all of these are exam questions. The first cell is in the DRG. The first synapse is in the dorsal column nuclei. The second order neuron is dorsal column nuclei. So the first was in the DRG, the second one is in the dorsal column nuclei. Axons of these nuclei cross to the opposite side, right? And then ascend in the medial lemniscus. medial lemnis. So the second order neurons goes up. It goes in the up through the medulla, pons, midbrain, then it reaches thalamus. So the second synapse is
Thalamus.
thalamus. The third neuron is in the thalamus. Neurons from here go to the cortex. So in the DCML system, we have at least 3 neurons. The first one is in the DRG, the second one in dorsal column nuclei, Grazian create, right? The third one is in the thalamus. In thalamus, we have a nucleus which is specifically for tactile sensation called VPN, ventral posterior nucleus. Ventral posterior nucleus. Ventral posterior nucleus. It has a lateral part, right, for collecting information from all over the body, unless the face, right? We have ventral posterior nucleus medial that receives touch and tactile sensation only from the face. So remember, the back of the head goes to the lateral part. It's together with the body. But the face is actually goes to the medial side, right? We have also inferior and superior. I don't want to talk about these now, okay? Let's talk about these. Lateral and medial, okay? So the lateral is from the body, the medial collects information from the face. Now let's see the orientation. I told you that in the DCML system, orientation of the fibers is very high. In anterolateral system is low. Orientation means the location or the relation between the fibers in the dorsal column. So you can see that fibers from the leg go to the gracile. That's it. But fibers from the hand go to the cuneus. Okay? So we have a range of the fibers in the cervical segments. Those that are located medially or close to the midline are from the lowest part of the body. Then, when we come up, fibers that are originating from upper and upper part of the body actually tend to be located laterally and laterally and laterally. And the most lateral one is fibers from the end and up. Okay? So if you cut the cervical section, the cervical segment of the spinal cord, fibers that are located medially are from the lower part. Fibers that are located laterally are from the lateral or the upper part. What does it mean for us? I mean, what is the importance of this? You may have a damage in the spinal cord, in the dorsal column. Okay? The damage may be localized. So you should know the consequence of that damage. If the damage tends to be located laterally, for example, in these areas, okay, you can think that maybe the sensory loss is in the upper part of it. If there is a tumor inside, I mean between the dorsal columns, okay, a pressing tumor between the dorsal columns on the two sides, then the pressure will be on the medial neurons, right? Sensory loss will be in the lower part of the body. Okay, I may ask something like this. Okay, there is sensory loss in the leg. Okay, the sensory loss is mainly for touch. There is no problem with pain. Okay, where do you think that we have a damage? Okay, the spinal cord that affect the dorsal column, from the midline. Okay? So this is the DCML. Again, the orientation changes from this point. So the blue one was located medially, the red one was located laterally. When the fibers cross, everything changes. So in the medial lemniscus, fibers change their orientation. They go up in the thalamus. In the thalamus, you have also orientation. Lateral receives from the lower part, medial receives from the face, right? Then, third order neurons that go to the cortex again change their orientation. You can see the red one goes to the lateral side, the blue one goes to the medial. So a stroke that affects the medial side of the cortex may cause sensory loss in legs. A stroke that may happen in the lateral side of the brain may cause sensory loss in hands or in face. Right? So this is the DCML system. So again, fibers go to the thalamus. Some of them go to the lateral side. These are from the ventral nucleus, ventral posterior medial nucleus for face. Then ventral posterior lateral nucleus send the fibers to the medial side and the medial part, right, for the body. Now let's see the representation of the body in the cerebral cortex. So the part of the cortex that receives the somatic sensation is called somatosensory area 1. It's located behind the central fissure. So from here we have frontal lobe, here we have parietal lobe, right? Somatosensory area 1, we call it S1, is located just behind the central fissure. In Brodmann's area is 1, 2, and 3. 1, 2, 3. The 3 is deep inside the fissure, right? Then we have 1 and 2. Okay? 1 and 3 is deep. Then we have 1 and then we have 2, right? So it's a gyrus. Somatosensory area is a gyrus, right? So we have some deep areas and some highest areas, okay? So Brodmann area 1, 2, and 3 account for somatosensory area 1. You have also somatosensory area 2, mainly in Brodmann area 42, okay? And some of them, actually, and some parts of the 40, okay? So, now let's see the orientation. Thigh is located medially, face and tongue are located laterally. Trunk is located in the middle part. Okay, so you should have an understanding of the brain. In the medial side we have legs, then we have hand, and trunk, and hand, and face. Right? So let's see this one. It's a half of the hemispheres, or one hemisphere, okay? So between the hemispheres we have sensations from the genital areas, then we have toes, foot, leg, hip, trunk, neck, right? And on the lateral side we have face, teeth, tongue, pharynx. In the middle part we have hands and trunk, right? So you can imagine that a person is lying on the cortex. He puts his leg between the hemispheres and the face is hanging on the lateral side, right? And on each side of the hemisphere, we have sensations from the opposite side. Sensation from the right side happens, left side happens in the right brain, and sensation from the right half of the body happens in the left hemisphere. Why? Thoracic the medulla. Very good. So, touch sensation in the spinal cord is ipsilateral, right? From any leg or any part of the body, fibers goes in the dorsal column on the same side. But at the level of the medulla, fibers go to the other side. So from this point, right, everything happens on the other side of the brain. Okay? So, what's the function of the cortex for touch sensation? So the function is revealed when you remove it. When you remove the part of the cortex. So, if you remove the somatosensory area 1, what happens? It mainly affects discriminative touch and position senses. For example, the patient may have agraphesthesia. Agraphesthesia means the patient is not able to understand what is drawn on his palm or any part of the body. For example, the experimenter may write letter A on the palm. Okay, the patient cannot feel the letter A. Or the experimenter may write, for example, number 2. The patient is not aware of that. The patient cannot feel it. Okay? The patient cannot say which letter was written in the palm. We call this agraphesthesia. Impaired ability to recognize letters and numbers drawn on the skin. Another symptom is astereognosis. Stereognosis means understanding the form of object by touch. You are testing that? Okay. So, for example, if you close your eyes and somebody puts something in your hand, by touch you can say the name of the object. For example, this is a cell phone, this one is a marker, this one is an eraser. Okay, you can feel it. You can understand and recognize the object. We call this stereognosis. When there is no ability for that, we call this astereognosis. So A calls the meaning of this one negative, right? Astereognosis means impaired judgment about the shape or form of the object. Another condition is hemihypothesia, radiosensitivity, sensibility to touch. There are also loss of vibration, proprioception, and fine touch. Localization is lost. So, a person with damage in the somatosensory area cannot tell the precise location of the touch. The patient is able to generally say that, okay, I feel it's on the right leg, but where in the leg, he or she doesn't know that. Okay? So, for example, you may know the precise location of the touch, but the patient is not able to do that. Okay? Localization is lost. Impaired judgment about the critical degree of pressure, if the pressure is high or light, the weight of the object, if it's light or heavy, the patient doesn't know anything about that. The texture of the material, for example, if it's wood, smooth, or glass, or plastic, the patient cannot do that, okay? So we can say that somatosensory area 1 is very important for all aspects of touch, pressure, and vibration, right? So let me get back to this slide. I told you that we have somatosensory area 1, somatosensory area 2, but there are 2 other areas for somatic sensation. Here, in Brodmann's area 5 and 7. Okay? So, we call these areas somatosensory associational area. So, 4 different areas for somatic sensation in the cortex include somatosensory area 1, somatosensory area 2, somatosensory association. You should know also the Broadman area. Broadman area 1, 2, 3, 5 and 7 for associational, and it's better to have a look on also on the Broadman area for somatosensory area 2. I think that it's 42 and 40, but try to make sure about that, okay? So, now let's see what's the importance of the somatosensory association of it. You know, in the cortex we have usually parallel processing and serial processing of information. Serial processing means any information is processed in some special areas in the brain, then the results go to another area, that area processes it further, then it sends the result to a third area. Okay? So processing happens serially. Some other time, some information goes to, for example, parallel areas. So two different areas process the information at the same time, but in different aspects. Okay? In the somatosensory cortex, information processing starts in the somatosensory area 1. Right? So when you touch an object, the basic feature of the touch is detected by the somatosensory area 1. Basic features are pressure, vibration, temperature, edges. So you can see the edge, you can feel the texture. So these are called basic features. So far, you didn't have any judgments about the nature of the object, right? These basic features, after the analysis, go to the association of it. So Broadman area 5 and 7 receives actually partially processed information from the somatosensory area 1, and they further analyze the results in order to judge about the form or the name of the object. So the final perception of object happens in association of areas, right? But the basic features are extracted by somatosensory area 1. Okay? So now, consider these processes, okay? So from VPN, fibers go to somatosensory area 1 for basic features. From here, signals go to Broadman area 5 and 7, association area. Some of the information go to S2. Okay? We ignore usually this one because it has a very complex function in touch sensation. So I usually don't talk about the somatosensory area 2. So ignore this. Okay? Let's stick to these two. Somatosensory area 1, somatosensory association. So this one is for basic features. This one is for complex features. Right? I told you. The form of the object is detected here, but edges, texture, temperature, everything about the object is detected here. So, if we remove this area, both basic features and complex features are lost. Right? Vibration cannot be detected. We have this in this slide. Vibration is lost, proprioception is lost, so the patient is not aware of the body part. Fine touch is lost, localization is lost, right? But also stereognosis happens, because for judgment about the form of the object, we need basic features. Basic features could reach here, and these areas analyze it for further judgment about the object, right? So when you have loss of this area, both basic features and complex features are lost. Now, you may have a patient that has an intact somatosensory area 1, but has a damage in the somatosensory association. What happens in this patient? You can expect basic features are intact, so the patient can feel, can tell the location, right? For example, vibration is okay, touch is okay. The patient is able to say the texture, the temperature. So for example, he can say that if this object is hot or cold, okay? But the complex feature is lost. The patient is not able to say the form of the object. A stereognosis again happens here, right? But there are also some other complex problems that the patient is unaware of the other side of the body, the opposite side, because the final sense of proprioception happens in the somatosensory association. So the position of the limbs, the movement of the limbs are finally judged in the association. Because the patient is not aware of the position sensation, he or she may not have any understanding on the opposite side of the body. So there's a damage on the right side, the patient doesn't have any understanding of the left side. We call this asomatogenesia. Actually, we don't have the word here, so asomatogenesia means the patient is not aware of the left side of the body. So this patient may want to get dressed, okay? He or she forget the left side. Asomatogenesia, yeah. So problem with body awareness. Okay, asomatogenesia. So the patient has problem with body awareness on the opposite side. So the patient wants to shave the face. He shaved the right side, forget the left side. Right? Or everything, okay? For example, maybe she's a lady, wants to do makeup, forget the left side. Okay? So, we call this asomatogenesia. Another condition is inattention to one side of objects. So, the patient is attending this object. Okay? Good. Imagine that you are the patient. Okay? With any special neglect. This one. So, the lesion is on the right side of your brain. You cannot attend to the left side. If I ask you that, what colors of the markers do we have here? You would say green, black, and blue. Okay? You won't tell the red. Why? Because we only attend the left side. Sorry, you only attend the right side. You forget the left side. Right? So lesion is on your right brain. You cannot attend the left side of the object. We call this hemispatial neglect. It doesn't mean that you cannot see this. There is no problem with vision. The only problem is that you don't pay attention to this. Attention is something different. Attention doesn't mean that you didn't, or you don't receive the information. But it means that you don't pay attention to that. Like, when you are attending to me now, you don't attend to that sound of that fan. Here? Okay, from the start of the class, we had this. But you didn't attend to that. Right? But now, when you are thinking about that, you know that it's there actually. Right? Attention is like this. So the stimulation is present, but the brain doesn't pay attention to that. So in this patient, the patient doesn't attend to the left side of the object. So we call this hemispatial neglect. Hemispatial neglect. I will see this in your final session. In your last session, I'm going to talk with a lot of details about these conditions. Okay? So, but for now, a patient with damage to the Brodmann's area 5 and 7, associational areas of the somatic sensation, has two problems. Right? Asomatognosia, also stereognosis, right? And hemispatial neglect. Collectively, we call this amorphosynthesis. Amorphosynthesis, right? So, it's about 10. Let's have